Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be starting with the topic of human gene therapy. So we will be covering the first part of this topic in this session. Okay, so before starting with gene therapy, I would just like to give you a brief overview or a brief definition of what is gene therapy. Now, what is therapy? All of we know. And what is gene therapy? Gene therapy means when we are utilizing or manipulating a gene as means of therapy to a particular disease. Now, it's not necessarily that that disease needs to be a genetic disease. No. Besides genetic disease, different other types of diseases can also be, managed and treated by gene therapy. Okay. Now, what is the basic principle of gene therapy? So here we are utilizing the gene as the therapeutic agent, isn't it? So this is the therapeutic transgene, for example. Transgenous specific sequences present on a particular chromosome, mainly on plasmids, as I've already discussed in the previous sessions. So, say for example, this is the therapeutic transgene that I have isolated or that I have obtained, and now I have to insert it into the target cell. Target cell means what? Target cell means that cell which have been affected by that particular disease, which have been infected or attacked by that particular pathogen. Okay, means the diseased cell. So say for example, this is my diseased cell. So obviously the diseased cell have a nucleus of its own. Now this therapeutic transgene will go and integrate itself with the chromosome or with the DNA that is present inside the nucleus of the affected cell. As a result of which, what will happen? The therapeutic transgene will be able to transcribe and then translate and finally produce the therapeutic protein, which will go travel outside the cell or which will travel outside the nucleus and lead to the treatment or the therapy of that particular disease. So this is the basic principle of gene therapy. Okay. Okay, so starting with the introduction, see, I have given a nice comic type of a diagram. You can see, you can say it's a typical comics type of a representative. So this is a DNA that we have, double helical DNA structure, and these two persons are basically playing or not playing, like they are basically doing some construction, some manipulation in the construction of these two strands by utilizing the or by changing the different base pair sequences that are there. Okay, these are the individual base pair sequences, kind of horizontal ladders. Okay, so gene therapy is the introduction of genes into existing cells to prevent or cure a wide range of diseases. As I mentioned, not necessarily genetic diseases, but also other diseases like infectious diseases. It can be cancer, it can be autoimmune disorder, okay, and obviously genetic diseases. So it is a technique for correcting the defective genes responsible for disease development. Because if we go into the depth and if we study, if we try to understand in details about each and every disease that is there out there, we will able to see some of the other genetic component or some of the other gene predisposition has to be there, okay, for almost any disease. So that's why gene therapy is something which can be utilized for any type of disease. So the first approved gene therapy experiment occurred in the year 1990. 1990 means around 35-36 years ago, on 14th September, when Ashanti De Silva, it was the name of that person, was treated for ADA SCID. What is ADA SCID? SCID is severe combined immunodeficiency syndrome. And ADA SCID stands for adenosine deaminase deficiency SCID. Means SCID which has occurred due to ADA deficiency. Okay. Moving forward. So see, in this slide, I have just given a short overview of the major milestones that has occurred in the history of gene therapy. Okay, so see, it has started right from 1989. So for the first time, adenovirus vector was uh, like discovered. So it was the first officially approved gene transfer into the humans, which was done by Stephen A. Rosenberg. Then in 1990, the next milestone comes, where again we came out with a retroviral vector, which was the first therapeutic gene transfer in ADA patients, means in adenosine deaminase deficiency patients. The next milestone is in the year 1999, where again adenovirus vector was used, but here it is a kind of a negative milestone, because it led to the death of a person in whom gene therapy was attempted using adenovirus vector, and the name of that person was Jess Gensinger. So that is a drawback. Now, because see, any new technique or any new drug or any new therapy, any new management technology, it doesn't mean that everything will be 100% successful or everything will be 100% positive, isn't it? So something or the other negative, something or the other unfortunate incident can happen. Okay, but we need to know about those uncertain or unfortunate incidents also. Then in the year 2003, again utilizing adenovirus vector, for the first time, China was the first country to approve a gene therapy based product for clinical use. Then the next milestone is in 2009, when again adenovirus vector was used for the first successful phase 3 gene therapy clinical trial in the United States. Then in 2016, again a retroviral vector was used, where Europe approves a particular gene therapy agent to treat patients with ADS skid, means adenosine deaminase deficiency skid. Okay. Then in August 2017, US approved again a particular agent for leukemia as means of like gene therapy. Okay. Then in October 2017, just 2 months later, US approves accelerator gene silcolilin for large B cell lymphoma, means again a cell gene therapy kind of a thing. And then in December 2017, again using AAV vector. AAV stands for adenovirus associated vector. Okay, so there US approves the first directly administered gene therapy, voretrigen Ripervocop, for retinal dystrophy. Okay, so retinal dystrophy is a condition where the cells or the tissues of the retina, they gradually start to be decomposed, or they start to be degraded. So that is retinal atrophy. Okay. Now, again, in this slide, I have given a nice pie chart which is explaining the different diseases which have been studied in gene therapy trials. So, see, maximum percentage goes to cancer, 66.6%. Okay, then the green part, this is the light green part. The light green part accounts for 6.3%, where we have the infectious diseases. Then we have the light blue part, that is this one. Okay, this stands for 1.8% for the neurological diseases like Parkinson's disease, Alzheimer's disease, etc. Okay, then we have inflammatory diseases, deep blue. So this is the deep blue one, which is only about 0.5%. Then we have gene marking, which is 1.7%, this one, deep green. Okay, then for the red color, red or maroon, we have the monogenic diseases, which is 11.5%. Okay, so this stands for the monogenic diseases. Monogenic diseases means genetic disorders which has occurred due to defects or mutation in a single gene. So monogenic. Okay. Cardiovascular diseases will account for 6.2%. So the violet one or the lavender one accounts for the cardiovascular diseases. Then we have the ocular disease. Ocular disease means eye related. Any disease which is related with eye manifestation. Okay. So that will be 1.3%. So this orange one is for ocular diseases. Other diseases will be 2.2%. Okay, this maroon. And healthy volunteers we also have around 1.9%. So this will be this violet, deep violet. They will be the healthy volunteers, means those persons who are not terminally ill but still they have volunteered for gene therapy trials. Okay. Okay, now again I have given in this slide a pie chart to make you explain the different vectors which have been used in gene therapy. So just let's see. So here we have the maximum portion is covered with a deep, more or less a medium shade of blue. So it accounts for 18.5% and it goes for the adenoviruses. Then we have the naked or the plasma DNA, which is 15.4%. It goes for this color of green, the light shade of green. Then we have lentivirus 9.5%, which goes for this shade of blue. Then we have pox virus 2.4%, which goes for the deep navy blue kind of shade of blue. Then we have different other vectors also, which accounts for 14.4%. This goes for the deeper shade of green. Then we have retrovirus 17.5% maroon, that means this shade of red. Then we have adeno-associated virus, that is AAV virus, 8.1%. I think it is corresponds to this shade of violet. Then we have vaccinia virus 4.5%. That is this orangish shade of orange. Then we have hub simplex virus, that is HSV, which accounts for 3.4%. It again accounts for this deep shade of maroon. And we have unknown viruses or unknown vectors also, which will be account for 6.3%. I think it goes for this shade of blue. Okay. Okay, now moving forward, we have different gene therapy trial phases. Okay, now what are they? Uh, like how will they be represented in a graph? Okay, so this is the graph that we have. Okay, so on the x-axis we have the number of gene therapy clinical trials, and on the y-axis we have the different phases. Okay, so let's just see. The phase 1 will correspond to around 56.1% of gene therapy clinical trials. Then see, gradually as the phase is increasing, because we know we have around 4 phases of clinical trials, isn't it? So as the number of phases are gradually increasing, the percentage of gene therapy clinical trials are also gradually decreasing. So at the last, at phase 4, we have only left with 0.2%. And then when it comes to the single subject, means where the subject has been finalized, okay, this will be the trial that will be like dealt with, then we have only 0.4%. Okay. Okay, now coming to the main content. Okay, so here as you can see, uh, gene therapy delivery can be of two types. One will be direct delivery and the other will be cell based delivery. Okay, so direct delivery means you are directly delivering the gene into the body of that person. But cell based delivery means you are collecting some particular cells or tissues from a particular organ of that individual, and then through those cells you are delivering the gene. That is the basic difference. Okay, so just let's focus on the direct delivery part first. So let's focus on the left hand side of the slide. So direct delivery में क्या हो रहा है? The cells are extracted from the patient, modified with the therapeutic gene and injected back into the patient. So say for example, this patient is having some problem with the liver, maybe fatty liver or liver cirrhosis or liver cancer, any type of condition, okay, dealing with the liver. So liver will be the target organ or target cell in case of this particular person or patient, isn't it? So in case of direct delivery, what we will do? We will extract some cells from the patient. Okay. So say for example, this is the therapeutic gene that we will be using in order to treat the condition of this patient. So we are directly delivering the therapeutic gene into a delivery vehicle such as a virus, because in most of the cases the vectors which we use are nothing but viruses. So say for example, this is the virus that we have chosen as the vector, and there we will be inserting the therapeutic transgene. Okay, so this is how it looks like. Then we will directly insert this therapeutic transgene containing virus particle into the body of that particular patient. Okay, but in case of cell based gene therapy, what is happening? So now let's just come to the right hand side of the slide. Okay, so here as you can see, the therapeutic gene is transferred directly to the target cells in the body. So in case of direct delivery, we don't know that whether that virus carrying the transgene will go into the liver or it will go into the heart or it will go into the stomach, because it is not a targeted delivery, it is just a random delivery. But in case of cell based delivery, we are directly targeting the transgene into the specific target cells. which in this case are the liver cells. So we need to isolate the liver cells first. We need to isolate the hepatocytes from this particular patient, and then we need to culture it in the petri dish. Okay, and then similarly we will be taking the therapeutic gene, just as in this case we have taken the therapeutic gene, we have inserted the therapeutic gene into the chosen virus or chosen vector, that is the delivery vehicle, and then we are putting this virus containing transgene into these plates together with the hepatocytes. Okay, so as a result, what is happening, the genetically modified cells are produced, isn't it? Because this transgene will go and integrate themselves into the chromosome of these cells, hepatocytes. So these will be the genetically modified hepatocytes which will be multiplying and replicating in the culture plate, because we have given the like optimum nutrients in the culture media. So see, these cells are genetically modified which are multiplying and replicating. And then we are taking these cells and inserting it or administering it or injecting it into the body of the patient. Now these are hepatocytes, these are liver cells. So these will only target the liver. There is no chance that they will go into the stomach or into the brain or into the heart. Isn't it? So that is why cell-based gene therapy is much more precise and concise method. Okay. Okay, now moving on. So in this slide, I have given a nice table which is explaining the different approved gene therapies that are there along with their delivery route and the vectors that have been used. Okay. So see, number one, we have FDA approved therapy. We have talimogene laherprepivec. So it's indicated in case of melanoma. Approval date is 2015. Delivery will be in vivo, means directly inside the body. And the viral vector will be HSV1, that is herpes simplex virus strain 1. Then we have Kimria. Kimria will be indicated in case of B cell precursor acute lymphoblastic blastic leukemia, that is all ALL. Again, approved date will be 2017, and also it's used in case of large B cell lymphoma, which was approved in 2018. Delivery route will be ex vivo, and viral vector will be lentivirus. Okay, so ex vivo means it will be outside the body. In vivo means inside the body. Ex vivo means outside the body. Then we have Yescarta, which is used for large B-cell lymphoma, approved in the year 2017. It is again ex vivo, and it is the viral vector used is a retroviral vector. Then we have Luxturna, used for mutation-associated retinal dystrophy, again approved in the year 2017. In vivo was the route of delivery, and viral vector was AAV, that is adenovirus-associated vector. Then the EMA approved therapy. Under the EMA approved therapies, we have number one is Imlygic, which is used for again melanoma, approved in the year 2015. Delivery will be in vivo route. Viral vector will be HSV1, that is herpes simplex virus 1. Then we have Strimvelis. Indication will be for ADA SCID, that is adenosine deaminase deficiency SCID, approved in the year 2016. Delivery will be ex vivo, viral vector will be retrovirus. And lastly, we have Zalmoxis, which is used for adjunctive treatment. Adjunctive means additional or extra. Okay, additional treatment in HSCT in high risk blood cancer, approved in the year 2016. So HSC stands for hematopoietic stem cell transplantation, which is very common as a means of treatment of blood cancer nowadays. So it will be done ex vivo, and the viral vector used will be retrovirus again. It cannot be seen. I'm sorry for that, due to the camera issue it cannot be seen. And so it's adjunctive treatment. And so some of these drugs are FDA approved, and while some of these drugs are EMA approved. EMA stands for European Medicines Agency. Okay. Okay, so moving forward, now there can be different approaches to gene therapy. Okay, like what? uh, first let's focus on the left hand side. We have gene replacement therapy, and on the right hand side we have gene addition. So talking about gene replacement therapy, so for monogenic diseases, again monogenic diseases means where only one gene, a single gene, is responsible to cause the disease, means mutation or any kind of change in a single gene has been responsible to cause the disease. So that means it's a monogenic disorder. So for such disorders, it involves replacing a mutated gene that causes disease with a healthy gene. So since only one gene is there, so if we can just replace that mutated gene with a healthy gene, then the purpose will be served. The purpose of gene therapy will be served, isn't it? So that will be known as gene replacement therapy. So instead of just calling it a gene therapy, we will be calling it a gene replacement therapy. Okay. So see what is being done. So this is the mutant gene, as a result of which the disease is getting caused. So we just take a functional gene and we just replace it with the mutated gene. Okay, or we take the mutant gene and we just form the correction that is needed. Okay, now coming to gene addition, just let's focus on the right hand side of the slide. Now here they are showing gene addition. So gene addition kab hoga? For complex and infectious diseases, means when it's not a monogenic disease, when maybe multiple genes are involved, mutations in multiple genes, or maybe various mutations in a gene is involved to cause the disease. So it's a more complex and complicated kind of a situation. In that case, we can introduce a new gene into the body to help fight a disease, often to supplement a targeted therapeutic agent. So we are additionally giving an extra gene, we are additionally providing an extra gene into the body of the patient with the hope that it will be acting as a supplement to cure the disease or to manage the disease. Okay, so here what is happening, this is the therapeutic gene, and we are adding this therapeutic gene to combat the disease or to alleviate the disease. Alleviate means to prevent or to manage the disease. So here there will be different genetic and environmental factors will be involved, which will be causing the disease. So if we can add a therapeutic gene, that can be helping with the alleviation of that particular disease. Okay, moving forward. So what are the different other types? So we saw gene replacement therapy and we also saw gene addition therapy. Now we are going to see gene inhibition, or which is also known as gene knockdown. Okay, means what? Means where we are carefully or deliberately we are switching off a gene, or we are inhibiting the expression of a gene. Okay, so inactivating a mutated gene that is overproducing its product by targeting the RNA. So say for example a particular disease is there which is happening due to a mutated gene, and not only mutation but also due to overexpression of that gene. So there is a gene that has become mutated and that particular gene is getting overexpressed now, and due to that the disease has developed into the patient. So in those cases, we will go for gene inhibition or knockdown. So see what they are doing over here. So this is the mutant gene that we are having, which is causing the disease. So we take a gene silencer, which will just go and silence the gene. Okay. So we have taken the gene silencer and we are putting it into here. So it is a knockdown. As a result, the disease is getting corrected, because the overexpression of the gene is no longer there, because the silencer gene that we have introduced inside the body of the patient has targeted the particular RNA which was produced from that overexpressed gene. So that leads to correction of that situation. Okay, so this can also be an option. Another option can be gene editing. So here what we are doing, we are just editing the sequences in the gene so that the mutation can be corrected, or so that the problem can be corrected. So it is making a targeted change to the gene sequence. What kind of targeted change? That will help in curing the disease, that will help in a management or treatment of that disease. Okay, so see again, we have the mutant gene which is causing the disease. So we take a gene corrector, it causes some editing, as a result of which the mutant gene is now corrected. Okay, so these are the four main techniques: gene replacement, uh, gene addition, gene knockdown, and gene editing. Four prominent techniques of gene therapy. Moving forward, so the aim of gene replacement therapy, or GRT, is to provide sufficient gene expression in enough targeted cells to ameliorate or correct the dysfunctional phenotype. Means to correct the phenotype means the dysfunctional protein, the dysfunctional hormone or enzyme which is causing the disease. So gene replacement therapy will be suitable for recessive disorders, single gene mutations, and X-linked diseases. Okay, X-linked diseases like we have sickle cell anemia, we have hemophilia, we have thalassemia. Okay, and recessive disorders means only one copy will not be enough to produce the disease. Both the copies of the genes, both the alleles need to be defective. And which it is unsuitable for which conditions? Dominant disorders where an errant gene codes for destructive or interfering proteins. If it's a dominant disorder, if it's not a recessive one, if only one copy of gene is enough to cause that disease, then for those cases gene replacement therapy will not be that much identical. Okay, and what will be the implementations? How can we implement this GRTs? So either it can be done directly in vivo, means directly put it inside the body of the patient, or through ex vivo cell therapy. Cell therapy means through by culturing, through the culturing of the cells in culture media, as I already mentioned. Or we can also use vectors which can be used to deliver the therapeutic transgene. Okay, now coming to the different viral vectors that are used for gene therapy. Okay, so there are basically five main classes of the viral vectors. What are they? We have retrovirus, we have lentivirus, we have Hub simplex virus type 1, that is HSV1, we have adeno-associated virus, that is AAV, and we have adenovirus also. So these are the five main classes of viruses which are used in the form of viral vectors for gene therapy. Okay, now retrovirus and lentivirus have some properties. What are they? They have integrating properties, so the genome integrates into the host genome. So this integrating property is not present in all types of viral vectors. Say for example, the other types that we have, HSV, AAV, adenovirus, these are basically non-integrating. So they will never go and integrate with the host genome. So they will persist in the cell nucleus predominantly as extra chromosomal episomes, means somewhat like plasmids. They will exist, they will not integrate themselves into the host chromosome, they will exist independently inside the nucleus of the cell. So this is the two distinct features between the different classes of viral vectors. Now the monogenic disease that we have, they represent 11.1% of diseases studied with gene therapy, with most gene therapy trials performed in the cancers. So most of the gene therapy trials have been performed in different types of cancers. Okay, now what are the different components of GRT, that is gene replacement therapy, when we are talking about the in vivo approach, means where we are directly injecting the gene into the body of the patient. Okay, so there are three main components of GRT. What are they? We have the viral vector, as you can see over here in the diagram. Then we have the transgene, this is the transgene, and then we have the promoter. And we also have the termination signal, which is also a component of the GRT. Okay, so what is the role of the viral vector? So it is the basically delivery vehicle. For the delivery, it is needed. So it is injected into the patient, and by which a transgene is delivered to the targeted cells of the patient. Then we have the transgene, which is nothing but a sequence of complementary DNA or cDNA, which is coding the replacement gene, means the corrected gene, which is actually required inside the body of the patient. And then we also have the promoter, which will turn on, it's basically the turn on switch. So as the promoter turns on, the transgene will start expressing itself. So it is the DNA sequence which acts as a turn on switch and modulates the expression of the gene. So promoter is something which I have already talked about when I was talking about modulation of gene expression, and in that topic when I was talking about the different operons, lac operon, TRP operon. In those sessions I have talked in details about what is a operon, what is a promoter, what is a operon, what is a operator. Okay, what is the repressor sequences, etc. These things I have already talked about in details. Okay, so promoter is usually always upstream, means to the left hand side of the main gene sequence, that is the transgene over here. Okay, and downstream of the transgene we have the termination signal. So what is the importance of the termination signal? It was to end the gene transcription. When the gene is already transcribed, it's the end of the process, then we need to switch off the entire system. So then the termination signal comes, and when switch on is required, then the promoter comes. Okay, and we also have inverted terminal repeats, which are abbreviated as ITRs, at either end of the cassette to allow for synthesis of complementary DNA. So this ITR, or these inverted terminal repeats, they will be present on either side of the entire system in order to allow for synthesizing the cDNA or the complementary DNA. Okay. Okay, now there are some requirements which are needed for approval of clinical trials for gene therapy. What are they? Number one, knowledge of defect and how it causes the symptoms. Means what is the actual genetic defect that has caused the disease, and what are the symptoms, what are the outcomes of that particular defect. Okay. Then what we need a good animal model, which is preferably a mice or a rat or a chimpanzee or a guinea pig. Okay, then we need to go for human study, human or mammalian study, where we can use chimpanzee or ape also as alternative, and we need to grow those cells in vitro, means under lab conditions. Then no alternate therapies or patients for whom existing therapies are not possible or have not worked. So these, any kind of loophole should not be there. Okay, and also we need to perform safe experiments. Safety, again, something I have mentioned earlier. I will be talking about the different societal issues of medical biotechnology also, that is included in this course. So there also I will be talking about the different ethical issues, the different safety issues. These are some of the things which we need to keep in mind, okay, whenever we are talking about some such novel, clinical trials or novel therapies. Okay, and then ultimately we have two main types of gene therapy. So we have the somatic cell gene therapy and we have the germline gene therapy. So somatic cell, now what are the basic differences? Because again, you think, I think I've told this before also, कि always if you explain, if you remember, or if you study something in the form of a diagram or a chart or a difference, it makes it easier for you to remember for a longer period of time. Okay, so somatic cell gene therapy means therapeutic genes are transferred into the somatic cells. Germline means they will be transferred into the germ cells. Example of somatic cell gene therapy will be introduction of genes into bone marrow, blood cells, skin cells. And example of germline gene therapy will be genes are introduced into the eggs and the sperms. So somatic cell gene therapy will obviously not be inherited to the next generations, but it can, the germline gene therapy can be inherited and passed on to the later generations. So at present, regarding the somatic cell gene therapy, all the researchers are directed to correct genetic defects in somatic cells. But in germline gene therapy, for safety, ethical and technological reasons, it is not being attempted at present. Means germline gene therapy is still in the research level. Okay. So I think that's it. We will be moving on shortly to the next session of this topic, human gene therapy. Till then, thank you. Goodbye. Please like, share, and subscribe ITLS Academy.